Allez, parfait. Je pense qu'on peut démarrer. Donc, au nom de toute l'équipe qui coordonne ces webinaires organisés par l'association AGIREmed, qui est un réseau afro-méditerranéen pour les agrobiosciences et l'environnement. Donc, nous nous joignons tous pour souhaiter la bienvenue au professeur Jaime Prohens pour cette 6e conférence virtuelle. Et encore une fois, on lui souhaite la bienvenue et nous le remercions pour avoir accepté de partager avec nous ces recherches que nous allons voir tout à l'heure, hein, donc, quand on va présenter le professeur. Donc, encore quelques mots concernant cette action pour les gens qui viennent de nous rejoindre pour la première fois, que c'est une action qui vient de différentes universités. Donc, c'est des universités qui ont tout d'abord œuvré dans un projet de type Erasmus+, Capacity Building, donc qui a pu monter un master dans le domaine des agrobiosciences. Et donc, les universités sont l'université de Toulouse, de Rabat, de Marrakech, de Ben Grir et de Tanger. Et encore une fois, on remercie AGIREmed et Aristide Boucaré de nous accompagner, donc avec l'outil webinaire de Zoom, et de nous apporter son expertise pour le bon déroulement de ces webinaires. Et aussi, on remercie notre ingénieur à l'ESAT, Véronique Pivon. Alors, aujourd'hui, nous accueillons Jaime Prohens de l'Université de Valence. Donc, on va voir tout à l'heure de quoi il va nous parler. Donc, pour résumer sa présentation, donc il va nous parler de l'intérêt et de l'importance de l'introgression génétique dans le domaine de l'amélioration des plantes et avec un focus sur l'adaptation au changement climatique. Alors, Jaime, tout d'abord, nous avons une belle histoire avec lui puisqu'il était et il est toujours un partenaire du projet Erasmus+ Mabiova. Et c'est lui, donc là, on a actuellement les étudiants des 3 masters qui vont suivre Jaime. Donc là, maintenant, ils vont vous voir, Jaime. Now, your students of Mabiova, they will see you and listen to you. They already watched you in many times regarding the videos that you already developed and the courses, online courses using Moodle. And I wish to thank you on behalf of Mabiova professors and Mabiova partners. And your courses have been well appreciated, and also all the quality systems that you introduced in these courses. So now they will interact with you and ask you maybe questions directly. So Jaime, so when we see his CV, it's very impressive when we see all that he done and all that he is still doing. So Jaime is from University Politécnica de Valencia since 2009. So he is a specialist of Solanaceae vegetables and crops. So he is a specialist of breeding, including assessment of the biodiversity, hybridization, molecular markers, genomics, phenomics, and many other omics. He has co-authored more than 150 papers in very high journals, okay? And also he contributed in more than 300 meetings and congresses. And also he's a researcher of practical and applied breeding, because he developed 5 varieties of vegetables. So in his research, he was able to produce and to select new varieties, in total 5 ones. And also he has a strong background in coordinating projects. So he has coordinated, or he was a principal investigator of over 50 research projects and contracts, including several international and H2020 projects. He also be involved in associations and also in other activities, because he was editor of plant journal of breeding, like Eucarpia and Frontiers in Plant Science. And he wrote books. And also during the period of 2004-2008, he served as the president of Eucarpia, of which now he is presently the scientific secretary. So I just want to mention also in this webinar that we are trying to build something in agrobioscience, and we are developing an association that we call AGIREmed, with some activities in to develop research around agrobioscience. And also, he is now the director of the Institute of Conservation and Improvement on Valencia Agrodiversity at UPV. And also is a member of the board of Spanish Society of Genetics. And in 2012, he has awarded the title of Doctor Honoris Causa by the University of Agriculture and Veterinary and Medicine in Romania. So another time we thank him and we welcome him warmly to our action, to our webinars. And I will try to be short to let him more time to present his work. And just to have an idea, until now, regarding all the webinars, we have more than 1,000 inscriptions. And most of them are young researchers, postdoc and PhD students, and also master students, and also teacher and engineers. So just at the practical level, so all your microphone and video are switched off. And if you want to ask questions to Jaime, please use the Q&R button. So you can, as the other webinars, you can put your questions and they will undergo a vote by the attendees. So the questions that will have many votes, they will be asked to Jaime, and we have, as usual, our professor, the Abdelaziz Mouni from the University of Rabat, that will handle this part of questions and will interact between you and Jaime. And also, if you want to ask your question, you can use the blue hand that you have at the bottom. We call it lever la main. Just click on it and we know that you want to ask question to Jaime. And also you can use converser. It's another button in the bottom. If you want to chat with between you and also ask some questions to the technical board, to Aristide, for example, if you have some technical programs. So I will stop here and I will stop to share my screen. And Jaime, you can start your presentation.
Merci, Simon and Tassis, and bonjour à tout le monde, à tous les amis de Mabiova et à tous les participants. Je suis désolé pourquoi mon français n'est pas très bon et pour faire une présentation en français. Je le ferai en anglais. So thank you again to all of you. Let's see, I'm trying to, okay, here I'm starting with my presentation. So thanks again to all of you and to Simon and Tassis. And my talk is about modern strategies for introgression breeding for adaptation to climate change. This is a subject that I have been working in the last years and in which we have developed a new approach, which I will present here. And also I will present an example of what we are doing in introgression breeding for adaptation to climate change in one vegetable species, which is eggplant, aubergine. Okay, so well, it is clear that there is multiple evidence that we are undergoing a climate change. So Despite the confinement and reductions of emissions due to the coronavirus crisis, the CO2 levels are continuously increasing. For example, last week, 2 weeks ago, sorry, we had a new record of concentrations of CO2 in the atmosphere. So for the first time in history, we arrived to 418 ppm. So here you can see the graphs in which the concentrations of CO2, which is a powerful greenhouse gas, are continuously increasing. And here we have the increases during this last year, in which we have a peak in June. So what we know is that CO2 is increasing continuously. There is no, the curve of concentration in the atmosphere is not going down. It's continuously increasing, and this obviously will have an impact on climate. Some of these impacts, we are already seeing them, but it is clear that there are different models, because it's very difficult to know what will be the climate in the future, but there are different models that show that mild to severe increases of temperature will affect productivity. So these are different scenarios. A moderate scenario in which we will see for 2100, we will see increases here in Morocco, Spain, France of 1.5 degrees more, to other worst scenarios in which we have increases of temperatures of around 5 degrees, which is a lot. Obviously, this will have a big impact in agriculture, and we have to be adapted to this. So there are several studies on the impacts on climate change in agriculture. Here we have some of Europe, in which we have some countries like the Nordic countries in different models in which the productivity will increase. Of course, the bottleneck for productivity in the northern countries is cold. If the weather becomes warmer, they will have better productivities. But in other places like the Mediterranean, the situation is quite different. So in most of the cases, the scenario is of dramatic reduction in changes in the Mediterranean region: Spain, Morocco, Algeria, and Italy, and all the countries of the Mediterranean. So here we have some examples of the increases of biotic and abiotic stresses derived from climate change. This means that there will be more problems of crop losses, like here, but also there will be an important impact in biotic stresses. There is a report from the European Crop Protection Association in which it says that due to the warming climate, one new pest is appearing in Spain from North Africa every 10 months. So the pests are migrating to the north, and this will mean that everywhere we will get more pests and new diseases that before we didn't have because of the climate change. So some years ago, we developed a new paradigm, which is not so new. We call it as a new paradigm, but it's not so new, which is using and tapping the wild for adapting crops to climate change. So our basic idea is, okay, wild species are adapted to grow in very stressful environments in some cases. So why not using the genes from the wild species for adaptation to climate change for breeding our crops to make them more tolerant or resilient to climate change? So this idea was already 100 years ago. There was one very important scientist, Nikolai Vavilov. He was a breeder in the Soviet Union, and he had he was one of the first and probably the most relevant one to realize that by going back to the wild species, going back to the old varieties, it was possible to find genes that could be used for adaptation to stresses, and in our case, for climate change. So Nikolai Vavilov, who unfortunately was murdered by the Soviet system in 1943, so he had an ambitious plan to end famine through the world. He wanted to use the new science of genetics to breed varieties that would grow where none had survived before. The key was a treasure trove of genes he was sure he could find in the unknown and wild types that had been ignored by our ancestors as they started farming more than 10,000 years ago. So the idea was to go back to the wild species to look for the genes that gave resistance and tolerance to stresses and use these genes. So based on the ideas of Vavilov, we developed this new approach. By the way, if you are interested, it's a very nice history with a sad ending, but it tells the history of Nikolai Vavilov. This is a very nice book, which I recommend. It's like a novel, but it tells the history of Vavilov, which in the end is a sad one because he ends dying in a gulag prison and he dies from hunger. So he fought against hunger and died of hunger. So what's known since long time ago is that during the process of domestication, when we look at the genetic diversity, we have the wild species that grow in multitude of stressing environments, which are very diverse. And during the domestication, a lot of diversity is being lost. And these materials are adapted to modified environments, environments that have been modified by humans. And from these old varieties and early domesticates, the breeders and the farmers have developed modern varieties which are adapted to environments that are very modified to avoid stress. Agriculture is trying to avoid stress to the plants that we are growing. The less stressed they are, the more they will produce and the better they will be for farmers. So it is known that many wild species are tolerant to stresses. For example, here we have Solanum chilense, which is a wild relative of tomato. The modern tomatoes that we eat and we buy in the supermarkets, they have genes from these wild species, which is able to grow in desertic areas. Not for the genes for tolerance to drought, but for genes of resistance to diseases. Here we have Solanum incanum, which is a wild relative of eggplant, which also grows in very desertic areas. And even for rice, which grows in flooded conditions, there are some species which are able to grow under drought conditions, like Oryza rufipogon. So wild species have been used for breeding. In the last 100 years, breeders have made crosses with wild species, the crops crossed with the wild species for breeding our crops and to obtain better varieties. When we go to the supermarket or to the shops and we buy wheat, we buy bread, the wheat that has been used for making the bread many times has introgressions from wild species. When we buy tomatoes, they have introgressions from wild species, and breeders have used a lot the wild species. So, for example, here we have in sunflower, in wheat, in potato, in peanuts, in tomatoes. So, wild species have been used for breeding for many traits, but mostly for biotic stresses. Almost all the uses of wild species in breeding have been for tolerance to biotic stresses. This is because when the farmers grow the crops and a new disease appears, then there is a kind of panic, and breeders are pressed to obtain resistant varieties. So what they do is they look for wild species which are tolerant to this disease, they make the crosses, and they develop varieties that bring the genes for resistance to these diseases. So this is the typical process, and it has been used a lot in breeding and in obtaining modern varieties. One extreme example of this is tomato. So the modern tomatoes have genes from 6 to 7 wild species for resistance to verticillium, fusarium, alternaria, staphylococcus, virus fusarium, tomato mosaic virus, tomato spotted wild virus, to many viruses, fungi, and bacteria. So when we eat a tomato, we are eating also genes from wild species that have been introduced there by crossing. So we think that, okay, wild species have been used a lot for disease resistance, but now we have a bigger challenge, which is climate change adaptation. So we think that we have to shift the goalpost from disease resistance to climate change adaptation. Climate change includes also disease resistance, but more things also. Abiotic stress tolerance. So how can we use effectively and more efficiently the wild species for adaptation to climate change? Well, the conventional approach is to use one wild relative for a specific trait. So we have one problem, one new pathogen. We look for a wild species which is tolerant to this pathogen. We cross them, if this is possible, and we make backcrosses, and we get a tomato or whatever species with a gene that gives resistance to this disease. However, this approach is one-pronged, so they go for one trait at a time. So what we proposed several years ago was a new approach called Introgressiomics. We are in the era of omics, so we invented a new omics, and it's a kind of preemptive breeding. So we are preparing for the future, not only for saving or solving the present problems, but also having a kind of materials which are prepared for the future, so that in the future we can have a quicker response to the challenges that we will have. So we defined Introgressiomics as the mass scale and systematic development of multiple plant materials and populations with introgressions of genomes from crop wild relatives into the genetic background of crops that may allow developing new generations of cultivars with dramatically improved properties, in particular adaptation to climate change. So this is a publication we made in Euphytica several years ago. So, of course, using wild species in breeding is not easy. It's quite difficult. So wild relatives have many undesirable traits. They have chemical and physical defense. Plants cannot escape predators, so they have to defend. They have low yield, a small fruit size. Many wild species are difficult to manage under domesticated conditions. When we try to grow wild plants, many times we have problems of germination, development, flowering, fruiting. Often there are crossing barriers between the crops and the wild relatives, in which it's very difficult or impossible to make the cross. We need a lot of time for developing varieties when making a cross with a wild species, because the hybrids normally look like the wild species. It's very difficult from a wild cross to obtain varieties that look like a modern crop. So when we cross with a wild tomato, until we get a modern tomato without a big fruit size, with high yield, etc., typically it takes 6, 7, 10 years, or even more. So protocols are also needed for screening for climate change traits. Climate change is a very complex trait. Crop wild relatives are less represented in the germplasm collections, so they are less accessible to breeders than cultivated varieties. The access to the germplasm is often perceived as difficult by breeders. Many times with modern legislation, it's very difficult to get germplasm from other countries. And information on already existing pre-bred materials is often missing or unavailable. There are few evaluation data on crop wild relatives compared to cultivated ones, and sometimes the taxonomy is very difficult. And the breeders, we are not a specialist in taxonomy, and sometimes we don't know which species we are working with. So, nonetheless, what's the approach that we propose? Well, what we propose is, well, these are the steps from the germplasm banks to use crop wild relatives, identify them, select the crop wild relatives to establish core collections, which can be for specific traits or in general without any specific traits, just to create materials, to hybridize them with the crop, to obtain interspecific hybrids, then backcross to the crop with selection, and to obtain populations, which we call Introgressiomics populations, which are populations that carry multiple introgressions from wild species. And these materials, of course, we want to use them for breeding and to introduce them in the breeding pipeline to develop, in the end, elite materials, cultivars, varieties, with introgressions from crop wild relatives. So what defines Introgressiomics is the scale, the integrated use of many disciplines, germplasm, genomics, wide hybridization, selection, phenotyping, and in the end, the final objective is to obtain elite material ready for breeders. So how can we identify the target wild species? Well, Harlan and DeWitt were two scientists that defined the gene pools. So we have a primary gene pool, which consists of the cultivated species and the wild ancestor in which crosses are easy, progenies are fertile, so there are no problems for making the crosses. Then we have the secondary gene pool in which crosses are more difficult and gene transfer may be possible, but with some difficulties. And then we have the tertiary gene pool in which the crosses are very difficult, the hybrids are normally sterile, and they may require special techniques like embryo rescue to obtain hybrids and to obtain introgression populations. So the identification of what materials to use, it can be done on genetic, phenotypic, evaluation, environments, origins, and gene pools diversity. And there are some identification strategies like the focused identification of germplasm strategy, in which we can look, for example, if we have a wild species that grow in desertic areas, probably will have tolerance to drought. So in this case, if we want tolerance to drought, we can go to wild species that grow in desertic areas. So then, when we have the wild species identified that we want to use, we have to make crosses, hybridization. And in the crosses with wild species and cultivated ones, there may be barriers to hybridization, prezygotic or postzygotic. But there are some techniques that allow overcoming these barriers in interspecific hybridization. For example, if there is lack of pollen germination, we can make treatments in the stigmas, style pollination, etc. If we have problems of development of the pollen in the tube, then we can make pollen mixture with compatible pollen. We can make fertilization in vitro. We can make embryo rescue, we can grow immature seeds, we can modify the ploidy level. Also, if we have hybrids sterile, we can use them as a female because normally the male sterility is more intense than the female one, or duplicating the F1 genome to establish fertility. So there are some techniques that can help us in obtaining interspecific hybrids. So many times, what we observe is this: the parents are very fertile, the hybrid has low fertility, but when we start the backcrossings, when we cross the F1 with the parents, the cultivated parent, in the first generation of backcross, second, third, fourth, the levels of fertility increase. So because we are recovering the genetic background of the cultivated species. This is a general trend. So once we have obtained the first backcross, then we can say that it will be really possible to obtain introgression populations. So if we have a sterile hybrid, one option is to duplicate the genome. Many times when we obtain tetraploids, the tetraploids, the fertility restored because the meiosis can be more or less regular because there is good pairing during the meiosis, and then we cross the tetraploid hybrid with tetraploid parent, and then we obtain a first backcross generation, which is tetraploid. So of course, we are not interested in tetraploids in most cases. So when we have the BC1, we have to come back to the diploid stage, and this can be done by anther or microspore culture. Then we have a diploid first backcross, which we can cross again with the diploid crop, and in the end we get crop materials with introgressions. So as I said, the fertility is restored during the backcross process, but sometimes we find the problem that there is no recombination between some parts of the genome of the wild species and the cultivated one. And so we have big chunks of the genome of the wild species that goes together with the gene or genes that we want to introgress. In sometimes it's not a big problem, like in tomato, in the chromosome 9 of tomato, most of the chromosome 9 of modern varieties of tomato is wild. But phenotypically, it has no big impact. In other cases, it has a big impact. And in these cases, this may represent a problem like the TY2 gene in tomato, which gives tolerance to one virus, but is linked to negative genes that reduce the yield and the agronomic properties of the crop. So in the end, one of the things that we can obtain are introgression materials. One of the most interesting types of introgression materials for breeders is what we call introgression lines collections. So by means of backcrossing and selection with markers, we can obtain materials like this. What does this represent? This is tomato. This is a population obtained by our friend Tony Granell, who also made a seminar some weeks ago. And the blue, these numbers are the chromosomes of tomato, 12 chromosomes. And the blue is the genome of tomato. So here we have one variety or one line in which all the genome is tomato except one part which is from a wild relative, which is Solanum pimpinellifolium. Here we have another line which has a different part of the genome. So in the end, we have a collection of lines, maybe 40, 50, in which we have the whole genome of the wild species represented in different lines. So if, for example, we know or we observe that this line is resistant to one new virus or has high tolerance to drought, so we know that in this part, in this genome part of the wild species, there are genes that give higher tolerance to drought. So these materials can be used very quickly by the breeders because we have a tomato genome only with a small part of the wild species genome. So this can be used very quickly in breeding. Another way to obtain materials with introgressions are multiparent populations. For example, if we make a cross of 8 parents to obtain a magic population, which is a combination of 8 parents, and one of them is a wild species, then we get a population of materials which on average has 12.5% of the wild genome, and we don't need to make backcrosses. There is also a possibility to obtain a magic population of wild species, which can be used to identify QTLs or genes important for agronomic traits or for adaptation to climate change, and then use them for backcrossing. So not only backcrosses can be used, but also multi-parent populations strategy to obtain introgression materials. Now, up to now, I have talked about introgression by crosses, but introgression can be also made by new plant breeding techniques. For example, SIS Genesis. In SIS Genesis is like genetic transformation in which what we use is a transgene or a gene that comes from a wild species related to the crop. So this could be made by crossing, but what we make is directly, we take the gene from the wild species and we introduce it with Arabidopsis or with other techniques into the genome of the crop. So of course, many people say, okay, this is a transgenic plant, and the legislation in Europe at least considers them as transgenic plants, genetically modified organisms. But in reality, the final product is not different to a conventional breeding technique. So in introgression breeding, the typical one, what we make is that we get a gene from the wild species and we make a hybrid. We obtain backcrosses, and in the end, what we have is that the resistant gene is in the same locus than the susceptibility gene from the cultivated species. So they are in the same locus. In SIS Genesis, the introduction of the gene is at random. So it will not be in the same locus. So this is a difference with the strategy based on crosses. However, there is a technique which is induced translocation breeding, which has been used a lot in cereals, in which after obtaining a F1 hybrid, so gamma radiation is applied, this breaks the chromosomes, and then we have translocations. So we have that the resistant gene is present in a different locus than the susceptibility gene. So there is no big difference between what is obtained here and what is obtained by SIS Genesis. Unfortunately, this strategy cannot be used in many countries. But also we can make nowadays introgression without making crosses and without introgression. It's a kind of paradox. By using CRISPR-Cas, if we identify the single nucleotide polymorphism or the SNPs that are responsible for the change of function of one gene that gives more tolerance or that gives resistance to one stress, so we can just edit the crop, and so we just copy the allele from the wild species into the cultivated one. And so with this, without any cross, we would have the gene and the advantage of the gene from the wild species in our crop. So ideally, for the breeders, what it would be interesting is to have large collections of materials, of introgression materials from different species. So for example, if we talk about tomato, to have introgression lines from different species from the primary gene pool, from different species from the secondary gene pool, from the tertiary gene pool, multiple wild species introgression materials, and all this data in integrated phenotypic evaluation, pedigree, and genotypic databases, so that we have materials and we have the information of these materials, so that breeders, when they need source of variation to breed new varieties tolerant to stresses, they have a repository where they can find exactly the line or the materials that they need, and they can take them and they can cross with their pure lines and to obtain varieties very quickly. So this is a general presentation of our approach, and now very quickly I will present you a case of how, well, first is the take-home message that introgression breeding can broaden the genetic base of crops by incorporating genes from wild species to the genetic background of the crop, and that Introgressiomics is a holistic approach, so it's a general one that can make a major contribution to adapting our crops to climate change, as well as to the development of new generations of resilient crops with dramatically improved properties. So as I said, this is the general part of my presentation, and now I will present you a case in which we are working, which is eggplant. So what we have been doing in these last 7 years in which we started this process. So eggplant, aubergine, you all know it, is susceptible to drought. While we have many wild species which are tolerant. Here we have Solanum incanum, Solanum elagnifolium, that are able to grow in very dry conditions. Here we have some wild species in the control and drought tolerance and drought conditions in which they have tolerance compared to the eggplant in which the plant is almost dead. So the genes of these species, which you see here, the tip is without wilting, while in the cultivated is wilted. So they have a lot of interest for adaptation to climate change. So what we did is to look for 15 species, wild species from the primary, secondary, and tertiary gene pool. You can see them here. So we have, this is primary gene pool species, but these are secondary ones, tertiary. So very different from, as you can see, this is eggplant and these are the wild species. Some of them are very different. So what we did with this is to select these wild species because their interest in breeding for adaptation to climate change, and we crossed them. We crossed them with 6 varieties of eggplant. So we made crosses with 6 varieties of eggplant of 15, 20 accessions of wild species. And we obtained hybrids with all the accessions of the primary gene pool with Solanum insanum. Also, we obtained hybrids with all the species of the secondary gene pool. So different species growing in Africa and Asia, and which are tolerant to drought. In some cases, for some varieties, we did not obtain hybrids, but we obtained it for other varieties. And for the tertiary gene pool, we obtained hybrids with 2 species, Solanum elagnifolium, which is high drought tolerant, and Solanum torbum. Of course, for obtaining the hybrids, we had to make embryo rescue. So we had to pick the fruits very early in the stage of development and to rescue the embryos to grow them in vitro to obtain plants. So here you have an example of the 2 parents, cultivated and wild, cultivated and wild. You see that the wild normally are very small, and the hybrids. The hybrids, you can see that most of the cases are small. So very similar to the wild species or intermediate. Here we have with Solanum elagnifolium. You can see the eggplant, the elagnifolium, and the hybrid, which is very similar to the wild species. This is very common because the genes of the wild species normally are dominant. But in any case, we obtained many backcrosses. So the hybrid, we crossed them with the cultivated parent, with all the primary gene pool accessions, with most of the secondary gene pool accessions. We also obtained first backcrosses, and also, to our surprise, with Solanum eleagnifolium, which is the first time in which the backcross was obtained between eggplant, which is an old world species, and Solanum eleagnifolium, which is an American species. So we crossed a species originally from America with eggplant, which is from the southeast of Asia. And we obtained backcrosses the first time. So here you have one example of fruits from backcrosses. Some of them look a little bit like an eggplant, but others have prickles. They have very strange shape of the calyx, many prickles, but some of them already start to have the fruit size of typical of the crop. So apart from all these materials, we selected 4 wild species for developing introgression lines. We selected Solanum incanum, which has a high content in phenolics and drought tolerance. Solanum insanum, which is this one, which is the wild ancestor of eggplant and has interesting domestication traits. Solanum dasyphilum, which is tolerant to spider mites and phenolics content. And Solanum eleagnifolium, because it has drought tolerance and also has some alkaloids of interest for human health. So we developed introgression lines. So we started in 2008, and this is the results up to 2016. So up to now, we have 51 introgression lines, which cover over 70% of the genome. They are not complete yet, but we are, it's the first set of lines, introgression lines in eggplant. And we tested them for drought tolerance in different places, in Spain, in Sri Lanka, in Cote d'Ivoire, and we found through several experiments that there was in a QTL in chromosome 3 for drought tolerance. So we have identified a genomic region in chromosome 3 that confers tolerance to drought in the genetic background of cultivated eggplant. So now we have materials of eggplant which have higher tolerance to drought, thanks to the genes from the wild species. So for Solanum insanum, it's also a complex program in which we started with 181 BC1 plants, which we selected with marker-assisted selection, and we obtained selected plants, then backcross again, genotyping, backcross, genotyping, etc. And now we have a situation like this in which we have already a very good set of introgression materials, and in which we will end up with a very nice collection of introgression lines. So we have all the genome of the wild species into scattered into different introgression lines. So during this program, we identified QTL for prickliness, the presence of spines. In eggplant, they are not spines. Botanically, they are prickles. And we identified a region in which there are, now we have 4 candidate genes for tolerance, for presence of prickles, and we are very close to cloning the gene for presence of prickles. So it's also a very interesting outcome of the use of introgression breeding. With Solanum dasyphilum, we made the same. We have practically clean introgression lines. And a very interesting thing that is also of great relevance for breeding is that we observed segregation for color. And for example, in the crosses with Solanum insanum, we have observed materials that have anthocyanins and chlorophyll, others which have anthocyanins but no chlorophyll, and also others that have no anthocyanins and no chlorophyll. So these materials, and also in the cross with dasyphilum and a white eggplant, we also obtained very nice segregation in the introgression lines. So we have been able also to identify the genomic region where the chlorophyll gene is, and together with another magic population, we are very close to the gene. So we hope that we will be able to clone the gene very soon for presence of chlorophyll in the skin. For Solanum elagnifolium, we are a little bit delayed compared to the others because we are in the BC4, but we also have a very nice representation of the genome of Solanum elagnifolium in the genetic background of eggplant. And what we have observed is that we have segregation for the root system. Some of the plants have very long roots because Solanum elagnifolium has very long roots that explore a lot the soil. So now we have materials of eggplant with this type of root system. This is eggplant. All the roots are very close to the stem. Here, in these materials with introgression from the wild species, we have roots that explore a lot the soil, much more. So they have higher tolerance to drought. So here you can see the difference between Solanum elagnifolium, which is high tolerant to drought. These are, you can see, of course, it has grown less than the control, but the tip is fine. And here you have eggplant, which is very susceptible to drought. The plant is almost dead. So the take-home message of our experiments is that wild eggplant relatives are an important source of traits and genes for eggplant breeding, in particular for tolerance to stresses. The development of advanced backcrosses generations and introgression lines is possible with many wild species, including the primary, secondary, and tertiary gene pool, at least with Solanum elagnifolium, and the use of high-throughput genotyping platforms facilitates the selection and speeding up of the development of introgression lines programs. So that's all from my side. Thanks for your attention. I have to thank my group in Valencia, a young and enthusiastic group. Without them, it would be impossible to make all these kind of things. And also to the Crop Trust, who funded the research in breeding, and the G2PSOL project, in which we are also doing work in this subject. So thank you very much, and I hope I have not bored you too much. And I look forward to your comments and questions and to your interaction. Thank you.
Thank you very much, Jaime, for this fantastic presentation that summarized very well what is introgression, and also with a precise example how it can be used to improve eggplant in this case. And I remember when we were in the Marrakesh streets and you found this eggplant wild relative in the street, and it was and after you show us that you use this species to improve, to introgress the domesticate one, and how it brings new traits for improvement. It means that just very close to us, there is a good genetic material that, and we can use these kind of tools to do. I don't know if we can here speak about fast breeding or no. Okay, so with these techniques of fast breeding, that we can go quickly and improve very important traits. Thank you very much, Jaime.
Thank you.
And now I will let Professor Smouni Abdelaziz to animate the question part. There is many questions. So we have, as I said, the vote system. So it will be easy to choose the best ones that, at least the one that had a lot of votes. Don't know if they are the best or not, but they had the consensus of many of the attendees. So please, Professor Abdelaziz.
Thank you very much, Mohammed. So once again, I'm very, very happy to be here and to have the opportunity to interact with all the participants. Thank you very much. So what I want to say to Jaime, I want to welcome him to this webinar, and I want to say thank you very much for what you have doing and what you are still doing for our students. So, you know, Jaime, you are very, as Simo said, you are very famous here in Morocco, and all Abiova students know you, and it is a very good thing that they can see you and interact with you. So, as Simo said, we have many questions, what it is very good, and I will ask them according to the vote. So the first question, which is a technical one, about Introgressiomic. A question from Fatima Zahra Brish, who asked if Introgressiomic can be applicable in plant species which genome is not entirely known.
Yes, of course. If you mean for the genome entirely known, the genome sequence, of course, it's possible. What, in fact, when we started with eggplant, we started in 2008, and the genome was not published until 2014. So what you need is a genetic map in which you have markers throughout the genome, and using these markers and the genotyping of the backcross populations, so you can select the plants that are more interesting for you. The first introgression lines were started and were published in the late 90s. So at this time, there were no genome sequences, and there were some genetic maps, but without much density. And even with this, if you have 3 or 4 markers per chromosome, you can start to do Introgressiomics to develop introgression lines. So yes, it's possible. You just need a genetic map.
Okay, thank you. We have now a direct question from the audience. So Mr. Diagadiouf, you can ask your question.
Thank you very much, and congratulations to the speaker. It was very, very good. I learned a lot. And I have just, and thank you also for the organizer, my friend Aziz Smouni. I am Jagaju from Senegal University of Cheikh Anta Diop, Dakar. My question is, are you planning to use speed breeding to speed up your
yes,
your work, please?
Yeah, speed breeding can be used in many different ways. It can be combining many different factors, or some can be just very simple. One of the ways in which we try to advance generations quickly is by growing the plants in small pots. In a pot of 1 liter, so the plant grows 20-30 centimeters, and the plant becomes stressed, flowers, and produces fruit. And in this way, you don't have to grow big plants, you don't have to wait much, and very quickly you have fruit. Also, another thing that we can do for speeding the process is to make embryo rescue. In the case of eggplant, the fruit takes a lot to get physiological maturity. And after, this may take 2 or 3 months until the seed is ripe. But after 15 days, you can, or less, you can take the fruit, you can rescue the embryo, and you can grow it in vitro, and very quickly you have plants. So yes, speed breeding is a clear objective, because the more generations that you can obtain in 1 year, the better. Everything goes faster. Instead of needing 10 years, maybe you can finish in 3-4 years. Yeah. And of course, there is the possibility also to use other type of agronomic techniques like changes of light, some types of fertilization, inducing some stresses that accelerate flowering. So, yeah.
Thank you. So the next question from Ali Boulerbah from Marrakesh, who asked if it is possible to develop plants with high capture and storage capacity of atmospheric carbon by using Introgressiomics, and if you can take in account the soil type and diversity in the plant breeding techniques.
Yes. Well, for the first question, theoretically, it may be possible if you have a wild species which has these characteristics. In practice, I think that we are still far away of this, because high capture and storage of CO2 is a complex process. There are some breeding programs that are aimed at transforming C3 plants to C4 plants, but this is requires modification of many physiological and biochemical aspects in the plant, and up to now there have been no success on this. By making crosses with wild species, I also think it may be difficult, because if you have a plant like eggplant, it's a C3 plant. All the wild relatives are also C3. And I really see complicated at present to increase the capture and the storage of CO2. In some crops, it may be possible if we have wild species that have a higher capacity of storage of CO2. But in the cases I know, is something which we think it's complicated. There are other traits that seem easier to target for adaptation to climate change. And regarding the soil types and diversity, of course, soil is one of the most important parts to be taken into account in breeding. And also, in fact, one of the important parts of breeding in some vegetables, like in eggplant or tomato, is in breeding for rootstocks. So these introgression materials and these interspecific hybrids can also be very interesting as rootstocks. Maybe we have some introgression materials with Solano melagnifolium, which are not very productive or have some characteristics which are not good for commercial market, but they may be useful as rootstocks because they have a powerful root system. So, of course, taking into account the soil types is essential.
Okay, thank you. Now we have a question, a direct question from Mr. Farahi. So please, you can ask your question. Maybe we have a technical problem. So maybe we'll move to Mr. Smouni.
Okay. So next question from Abdessadek Boujaj, who asked, sometimes wild plants have genes for monogenic and even polygenic resistance. How easy it is to introduce a polygenic gene?
Okay, I think that the question is, how easy is to introduce a polygenic trait? Because a polygenic gene cannot be polygenic. So polygenic means several genes. Okay, but I understand the question. So of course, introducing a monogenic trait that depends on one gene is relatively very easy, because it can be done with a few molecular markers or just one molecular marker, and you can make the introgression. With polygenic traits, with many traits, then fortunately having molecular markers, if we have the genes identified or QTLs identified, of course, we need higher numbers of plants in the segregating generations. But if you have enough resources, and you have you can put many plants and you can genotype them, theoretically you can introgress polygenic traits or many different genes in which you are interested in the genetic background, unless they are very linked and there is few recombination in the area, and you have one gene of interest in the wild species and another of interest in the cultivated one. So if there is linkage drag, sometimes you can have problems for this. But introducing polygenic traits is feasible, and it's done with molecular markers, of course.
Next question, Simo. Simo? So maybe there is a problem. So maybe we'll pass the next question here. A question from Reda Ben Mrid, who asked, wait. A question concerning post-transcriptional and post-translational modification. And he asked if you, did you compare them between the wild type and the transformed species?
Yeah, we have not studied them directly, but of course, when you introduce pieces of a different genome into the genetic background of the crop, there are many changes in the physiology, and also in the there are modifications, transcriptional and post-translational modification. But from my side, I think that it's more important the level of the changes of methylation in the genome. So there are many epigenetic changes. This has been seen when we obtain hybrids. There are many differences in the methylation patterns. There are some regions of the genomes that are methylated, others which they were methylated, now they are not. And there are differences, important differences in the expression of genes. In fact, some months ago, we submitted a paper with some Italian and British colleagues in which we found that the vigor of hybrids, even intraspecific hybrids, can be due mostly to epigenetic changes. So the pattern of methylation is very similar to the very vigorous plants. So yes, there are many changes in which we do not know much about them. For sure, there is work for decades to study what happens in these cases.
Okay, now we will move to Mr. Ferahi. I think now his microphone is okay. Mr. Ferahi, you can speak. Well, I think
okay.
We still have a problem. So I will ask a question if Professor Jaime agree.
Yeah, of course.
Have you an example of an introgression that allow to select a population of microbiota that interact with roots? Is there any example for this?
As far as I know, not. But in our case, we have many hopes on this, because this wild species, Solano melagnifolium, it's very efficient in the absorption of nutrients. And this species has mycorrhiza. So we are interested in identifying if there are some genomic regions in this wild species that gives the ability to interact with mycorrhiza. So it's one of the things that we plan to do in a near future. Of course, this would be very interesting. I don't know if it will be easy or not. I guess it's not a thing of one gene. Probably it's a more complex inheritance, but we'll try. It's one of the mycorrhiza give higher efficiency in the use of water and in the use of nutrients. And this is very interesting.
This will be fantastic because, yeah.
It's very interesting for adaptation to climate change, of course.
And also it can maybe bring new knowledge how genes can select microbiota and also at the fundamental level to go to the mechanism and also the pathway. This will be fantastic. Okay, Monsieur Aziz.
The next question from Professor Leila Mdraoui from Rabat, who thank you, Jaime, for your interesting presentation, and who asked about crop wild relatives based Introgressiomics. Can they be considered as a GMO?
Thanks for the question. No, because introgression breeding based on crosses is considered as a natural technique. So most of the crops that we are eating every day, they contain introgressions made by breeders through crosses. And this is one of the big advantages of this approach to use wild species, is that there are no absolutely any concerns or special regulations to grow them. So it's a big advantage according to the European legislation. Of course, we are aware that crossing with wild species can bring some surprises and not always good ones. If we cross with species that have high levels of alkaloids, wild species of tomato, of eggplant, they have to survive against predators. And one of the ways is with alkaloids. They are very bitter and they can also be damaging for health. So if when we cross and we also bring the presence of alkaloids, it can be, we can obtain a variety which is poisonous. And of course, we are looking at this and we are analyzing the alkaloids in the introgression materials, because this would be a disaster from the public health point of view. But the great advantage of this technique is that there are absolutely no legal problems on marketing them. They are not considered GMOs.
Okay. And Jaime, how you check that? You do metabolomics or you do some test on animals to give them to eat this? Okay.
We make analysis with HPLC or any platform or metabolomic platform. Yeah.
Okay.
So next question, I will join two questions which are going to the same way. So from M. Baziz and Fatima Zahra Brish. So since gene transfer by crossing take more time and is facing infertility, can you use instead mutagenesis basis or chemical and physical treatments? And if you can give a comparison. And the other question, what is the easiest method to be executed by the breeder to make cross or to introduce genes responsible for resistance directly to a genotype?
Okay, so mutagenesis is a very powerful technique and it has been used a lot on breeding. But when you introduce mutagenesis, you normally, what you do is you knock out genes, basically. So yes, you can obtain maybe materials that are more tolerant to drought, but normally they have also, apart from this tolerance to drought, they have also other phenotypes with less production or lack of chlorophyll or other traits that normally are associated to these mutations that for breeding are not very good for some traits. For drought tolerance, there are many genes which are known, but in general, the approaches using mutagenesis, only mutagenesis to obtain tolerance to drought have been not very effective up to now. So, of course, if you have directed mutagenesis with CRISPR-Cas and you know the gene that gives tolerance to drought and you modify it, you could get advances. But by using mutagenesis with chemical or physical agents, the results have not been very promising so far. And the other question was, what's the easiest technique? Of course, if we know the gene that we want to introduce, we know its sequence, we can do CRISPR-Cas and to change the gene of the cultivated species to the gene of the wild species. We can make a copy of the allele of the wild species into the cultivated one. But this cannot be used up to now in Europe. In other countries, yes. If we have to use techniques that are legal in Europe, nowadays it's crossing and use of molecular markers for marker-assisted selection.
Thank you, Jaime. So next question from Omar Idrisi. From Omar Idrisi, who asked when using only wild species from primary gene pool in interspecific crosses, could we expect satisfactory results in terms of abiotic, biotic resistance, or should we go further and use secondary and tertiary gene pool? And one other related question from Mary Broad: how do you characterize the primary, secondary, and tertiary gene pool from for the wild relative species?
Okay, so for the first one, of course, breeders, I would not say that they are lazy, but they try not to complicate their lives. If we have the genes that we want in the primary gene pool, of course, the most satisfactory strategy is to use the primary gene pool. But many times the gene pool is the wild ancestor. And the wild ancestor, many times, the genetic diversity that we find in it is not so high. So many times in the primary gene pool, we don't find very good levels of tolerance. For example, in tomato, the wild ancestor is Solanum pimpinellifolium. And in Solanum pimpinellifolium, yeah, you have some tolerance to drought, but you have other species from the secondary gene pool that grow in desertic areas that are very tolerant and resistant to drought, and they are much better to get tolerance to drought. So yes, if you have the tolerance in the primary gene pool, it's better to use the primary gene pool. If not, in the secondary and tertiary gene pool, you normally have much more genetic diversity for choosing the wild species that you want to use. And the other question was how we characterize the primary, secondary, and tertiary gene pool species. So this means how we classify the wild species in primary, secondary, and tertiary gene pool, or how we make the characterization for drought tolerance. Well, if this is the first question, then classifying them in primary, secondary, and tertiary gene pool species is by means of crossings. So if the crossings are easy, it's primary. If not, they are tertiary. If they are intermediate, secondary. Sometimes there are species which have not been tested. In this case, you use the phylogenetic tree to classify them in primary, secondary, or tertiary gene pool species. If what you are referring is to how we characterize them for drought tolerance, normally what we do is a first screening with plantlets, and we see how they respond to drought. Normally, there is a quite good correlation. If they are tolerant in the seedling stage, in the adult stage, they are also tolerant. So we just subject them to the same treatment of drought without watering, and we select the best ones.
Okay, so I will ask one question from the direct section. Jaime, you present to us all this collection that you have, that very interesting collection to select traits and new lines. So I mean the magic collection and introgression collection. So if I ask you just one to give us an estimation about the time, so how long it can take, for example, to select high phosphate or low phosphate tolerant lines with some introgression from the beginning of the experimental design until to have the line to the market?
For traits like the one you mentioned, if we say, okay, we have this species which has high tolerance or has high efficiency in the absorption of phosphorus, and we have to start with the crosses and to develop the introgression lines and to test if they are if they have the trait of high absorption of phosphorus, and we have to cross them with lines of the breeding companies and to obtain varieties. It can be perfectly 10, 12 years.
Wow, okay.
Nowadays, even using speed breeding techniques. Maybe with speed breeding is 7, 8 years, but still it's a long time for complex traits.
And just for the first part, to identify the gene and to try to understand how it works, maybe it's
five
five
years.
Okay,
yeah.
It's a PhD.
Yeah,
it's
a long PhD.
Okay. A long PhD. Okay.
Thank you, Jaime. So next question from Professor Zinlaibidin Triki from Rabat, who want to thank you for your presentation and also for courses in Moodle, Mabiova Moodle. So he's one of the coordinator of the master. So his question is, you described breeding for problems of climate change in Mediterranean context with droughts and rising temperature. And he asked if there any work on introgression of other traits in other geographic contexts, for example, immersion or others.
Yeah, for flooding, when there are flooding. I know that there are some of these works on trees. In trees, introgression breeding is very complicated because it takes a lot of time. But here in Valencia, there is works since the 60s and 50s in which they were interested in rootstocks. And one of the problems that sometimes we have here is flooding. In autumn, we have many times very big rains, and you have the fields flooded for several days or even weeks. And this is very stressing for the plants. So yes, there are some works on adaptation to flooding. Of course, when you have introgression lines, you can test them for drought tolerance, but also for flooding tolerance. So yes, there are works on tolerance for immersion.
Okay. So we are almost at the end of our webinar. So maybe we can take last question, Professor Aziz.
So if Jaime agree, I have one question before. So I was interested when you are using interspecific crosses, and after you say that's in the next generation, when during the Backcrosses, the fertility is rising. So is there any similarity with the natural evolution of plants and the natural adaptation of the plants?
Yeah, I think so, because in fact, now in which Simo probably knows very well, in which now that we have the power to sequence the whole genomes, many times there are surprises in which you say, okay, this old variety of eggplant or tomato in which nobody used interspecific crosses has introgressions from wild species, so that have been produced during the evolution in which there was a cross with a wild species, and some parts of the wild genome are present in the cultivated one, and it was not intended. Nobody crossed them artificially. It has been just during evolution. Many times during the evolution of crops, the introgression with the wild species has been very important, because many times there have been unintended crosses that have brought diversity and also traits. And in the modern crops, we have introgressions that from wild species that we didn't know that were there. We assumed that there were no introgression with wild species, and there are.
Okay, thank you. So the last question, Simo?
Yes.
One more question from Professor Mohammed Azqor. And he asked, we are now sure that climate change is due to excessive and uncontrolled industrial activity. Within the intervention of researcher via transgenic and introgressiomic, is that we will not cause any damages in natural evolution in plant species?
Well, the first one, we are not sure about climate change. I don't know, because I'm not an expert in climate science, and I'm a breeder and geneticist. But in any case, the materials we are obtaining, materials that are tolerant to drought, generally means that they are more water use efficient. So they are able to grow with less water. Also, many materials are able to grow with less nutrients. Let's say that one of the objectives is also breeding for resilience, in which you have plants that are able to cope better with the stresses. So if we have plants that are able to cope better with the stresses, we have plants that are able to grow in places where they could not grow before. So in the end, we are improving agriculture. And if we are affecting the evolution of the plants, yes, of course. Cultivated plants are natural monsters. So they have been shaped by humans and the natural selection, natural and artificial selection. In general, if we leave a cultivated plant without the care of humans, they disappear. They are unable to compete with the wild species. So of course, breeding is modifying evolution, and when you cross with wild species, you are modifying the evolution of the species. In sometimes we, the humans and the breeders, we have created new species. For example, the triticale. Triticale is an artificial a hybrid between rye and wheat. And it's an octoploid that has been created by humans, and it's grown a lot in the center of Europe and the north of Europe. So yes, we are changing the evolution. Breeding is evolution under the control of humans.
Sure.
And in many cases, we are just playing the role of the bees.
Yeah.
Just to bring close to each other plants that are distantly separated. So
and helping the hybrids to survive and to be able to cross. But in nature, this happens. Nature has hundreds of millions of years in which whatever could happen many times has happened.
Yes, exactly. Many possibilities. Quantum field of possibilities, yes. Okay, so with these nice words, we wish to thank you another time, Jaime, for these very nice presentations and that very warm exchange at the end, with the very interesting, with this technique that is widely used, and you show us in a very nice example how it can change things and to do innovation in the context of climate change. And as you said, yes, it's important, and also something that we didn't say, it's also contribute to fight against hunger. We will be 9 billion soon, so we have to prepare this, and these techniques, that can select new cultivars, new varieties with high yield and also quality. Okay, and just before to close, just if you can stop your screen sharing, I want just to introduce the next speaker of the next week. Nice. Okay. So our next speaker is Antonio Monforte. So it's a colleague of Jaime. So he's from another institute. He's from the CSIC Institute of Valencia. And Jaime is also a genetist, and he works on many crops. And he choose for us another topic, because human, compared to animals, human, they like to eat food with good nutrients, but also food with good color, okay, and also food with different shape. So when you go to the market, you have fruit with different shape, different color, and different quality of nutrient. And Antonio Monforte, he worked among his subjects, he also worked on the shape, and he's interested on identifying genes that give different shape of melon, for example. Melon is just an example of his presentation, and he will tell us how he did, which approach he used to identify the genes that make the melon elongated compared to the melon that is round. Okay, so we know in Morocco there is many, and also Spain, we have similar melon in the two countries. So within this example, you will see how genetics can help to identify this loci that bring another trait that is also for us important. Okay, so with this, I on behalf of all the management committee and also all the attendees that follow us every week, to thank you another time, Jaime, and also we thank all the attendees, and to Aristide from AGIREmed to help us to conduct this webinar. And Jaime accepted to have this webinar recorded, and we will have it soon on our platform, so you can follow it.
Thank you.
For our students, it's important to, if they want to follow it in another time to better integrate all the areas that have been covered. Thank you again, and see you next week. And thank you again, Jaime. Ciao.
Thank you.
Bye-bye.
The webinar of Tony Monforte will be very interesting for sure. So I recommend it also.
Thank you.
So just to record the date is on Tuesday, and at exactly the same time as now. It means 4 o'clock in Europe and 3 o'clock in Morocco. and it corresponds to 2 o'clock of GMT. Bye-bye, everybody.
Okay, bye. Thank you. So bye, Assis.
Goodbye, Jaime. Thank you very much.
Bye. You're welcome. Thanks
very
to
nice
you.
to have you and to see you again.
Bye. We keep in touch.
Yes, of course.